being back with us here today, and in this fitting lecture theatre, of course, as well. So today, Brian will be talking about the language of living processes, genes, development, and embodied meaning in organisms, developing a postmodern science of qualities. Brian.
Well, thank you very much, Tom. It is an enormous pleasure to be back here. This is where I started my teaching career in 1965, in this very lecture theatre. So this is very familiar, and John Maynard Smith is extremely present here for me, and I'm sure he is for many of you. I'm rather interested in the composition of the audience. How many of you are biologists? And how many of you are cognitive scientists or robotics people or So that seems to account for most of the population. Physicists, mathematician? Yeah, okay. Very good, thank you. Just to get an idea of the composition of the audience, because I'm going to try to bring together a number of different areas, as you can see from the title. It's rather a long title. But what I would really like to do is two things. The first part of the talk is really science, and then I want to explore the implications of what I see as a new science and a new biology for the issue of education in general and how we relate to nature, because that's one of the critical aspects of the time we are living through. Exciting times. So I want to start off with this sort of crisis of the genes that came up with the Genome Project. And let me start with a quotation from Helen Pearson, who was writing in Nature in 2006. And here's Helen's reflection on the concept of the gene. Where the meaning of most four-letter words is all too clear, that of gene is not. The more expert scientists become in molecular genetics, the less easy it is to be sure about what, if anything, a gene actually is. Now, how many of you biologists would agree with that statement? Or you can agree up to 10% or 50% or 80%. Yeah, a conditional, there's a sort of qualified agreement with that. But you can see the point she's making is that as the concept of the gene has become more and more fuzzy due to the extraordinary variety of editing processes that genes go through, and the different ways in which genes are transcribed and their products are produced, it becomes less and less clear what a gene actually is. And so what I'm going to be doing is exploring this issue from the point of view of embedding genes in networks, which are the regulatory networks that make meaning out of the information in DNA. Now, meaning is not a word that is usually brought into biology, but I'm going to use it as primary, and I'm even going to use the term embodied meaning. And at the ECAL meeting, the European Congress on Artificial Life in Lisbon last year, I had the delightful surprise of realizing that there is a great deal of interest in embodied meaning in that community. And I realize this is Francisco Varela's influence, as well as the influence of many other people. And so the Maturana Varela view of organisms that they presented in the Tree of Knowledge is something that has had a great influence, I think, on the thinking of cognitive scientists. And it's recognized that most of the knowledge we have is embodied. It's not accessible to consciousness. But it's embodied in a form that results in coherent behavior and in meaning. Now, meaning is often associated primarily with the rational aspect of the signification. But I'm going to use meaning in the double sense of both signification and mattering That is to say, what the affective aspect of meaning. It has two sides. It has a logical aspect and has a more emotional or affective aspect. And I'm going to use both. So, let me give you a quotation that has come from Evelyn Fox Keller and David Harrell in a paper 2007, so last year, the Public Library of Science, PLOS, which is an online journal. And here is a statement that they make, because what they're doing is, as a result of this, whatever you want to call it, this difficulty of understanding how the information in the genome is turned into the coherent embodied meaning of the organism, which is development. And that was, of course, my subject throughout my university career from the 60s, the early 60s, through to now. So they're reflecting on these transformations that have occurred. And, of course, Evelyn Fox Keller wrote a book called The Century of the Gene, which I think was very full of insight about the issues that were coming out of the Genome Project. And here is Fox Keller and Harrell. From a logical point of view, the organism in its environment can be viewed as a universe of discourse, something logicians call a model or a world. Now let me just ask you, how many people does that make sense to immediately? No. Right, okay. It's a bit abstruse. And what I would like to do is try to unpack some of that. Because actually Maggie Bowden and I think she's still associated with the university in cognitive science, we wrote some papers that explored this territory years and years ago. This is in the late 70s. And I just find it interesting the way cycles of cognition and understanding do come round again. And what I think we're seeing is a re-emergence of ideas that were actually fairly, they weren't prominent, but they were present in the 60s and 70s. Can I just check on audibility? Can you hear me all right? So the ear system is good. Okay, now let me go into this, the more scientific aspect of my talk, the language of living processes, or the complex language of living processes. I'm introducing this idea of ambiguity in cellular regulatory networks. Now, ambiguity, again, is not a word that would be used by molecular biologists because it is assumed that the interaction of molecules within organisms is essentially deterministic. There's molecular determinism and causal determinism. So molecule A interacts with molecule B in a perfectly well-defined way, and the result is some causal effect, C. And you can go on with this chain of causal effects and describe them. And what is then often the case is that we attempt to model these processes in terms of what I'm calling here conventional model building, and that's what I did most of my career. Conventional model building in which we assume molecular determinism of this kind so that you can construct models in terms of differential equations, so these chains of causal processes. And you put them together in various ways with feedback loops and coupling to various types of activity, and we did this in morphogenesis to stress and strain tensors to get the shapes and forms of organisms in response to dynamics. And that kind of molecular modeling is something that is based upon molecular determinism, and hence differential equations, partial differential equations, and this kind of computer modeling. And again, I think that's familiar to nearly everybody in the audience. How many people would say that they have engaged in this kind of modeling activity during the course of their careers? Yeah, I mean, it's a very standard sort of process, so it's quite familiar. And as I say, this is what I did for much of my biological career. Now, complex systems with emergent properties is what I learned about at the Santa Fe Institute in the 80s and the 90s. And it was a bit of a revelation because of the realization that these complex systems can behave in ways that are really unexpected. And emergent properties reflect the expression of the whole complex dynamic system. And small changes in either the composition of the complex system or the patterns of interaction can give rise to totally unexpected emergent properties. Now these emergent properties are they're holistic features of the complex system, and they do reflect the aggregate dynamics of the process. I mean, you think of voids, these flocks of birds, of social insects, and I know a number of you are modeling these types of process. So what you do is you define the interactions between the constituents, and then you just give them simple rules of interaction, and then look at the behavior of the collective. And you find some very beautiful and often some rather counterintuitive things. Now, these are the sorts of models that I've worked with. I mean, I mentioned social insects, ants, termites, order out of chaos, long-range spatial order. Now, long-range spatial order is something I'm going to keep coming back to. Long-range order that is actually a result of a particular type of dynamic and network property. And it's very closely connected with fractal structure, self-similarity in the interaction networks that are involved in these complex systems, complex dynamic systems. And just to give you an image of that, we have here, can you see that all right? Okay, that's obviously a termite colony. And if you look at the dynamics of the termites, you think they're just behaving chaotically. You know, they gather together in these groups. Suppose they're repairing a part of it or extending the building, this colony. And they gather in groups. They have, of course, pheromones. They have these little pellets, fecal pellets that they use for building, which is dust and their own feces, filled with pheromones. It attracts other termites, so that you get clusters of termites of all sizes that are working on the construction of the pillars and the halls and the different parts of this architectural marvel. And then suddenly they freeze, and then they scatter. And you think, what the hell is going on here? These guys are just doing as they please. They do a bit of work, they do something, and then they stop and they scatter and they go somewhere else. And it turns out that when you do the sums, the mathematics on this, then you find out that in fact they're doing exactly what they should be doing in order to produce coherence over the whole of the construction site that they're engaged in, and nobody is telling them what to do. So that this fractal dynamics, this self-similar dynamics, because the clusters on all sizes is actually producing long-range order and coherence. Now just imagine us engaging in that kind of architecture. We always do a rational top-down design of architecture. I mean, we're always in our culture, in modernity, we do. In traditional architecture, of course, there's plenty of this organic construction going on. And then you get self-similarity of cities, you get self-similarity of villages, you get self-similarity in the growth of these self-organizing communities. So humans also engage in this kind of self-organizing. Wow. Okay. Well, I will not touch him again. So this kind of self-organizing activity is also something that humans engage in. But we in modernity have tended to focus on the rational aspects of this and order and control, in particular control. So that's what this is all about. Not the termites. They're also controlled, but they're also cooperating. So here are these networks of interacting units with fractal properties. Now let me just get into fractal structure a little bit here. Again, I think this is familiar to nearly all of you, but it's showing up over and over again in different aspects of these biological systems. And the metabolic network, for example, is fractal in its structure. Protein interaction network, genetic regulatory network, I'll go into this in a bit more detail. Heart rate variability, which is quite interesting. Heart rate variability has a fractal structure, chaotic component. Chaotic attractors, as you're well aware, are fractal. They're self-similar in their pattern, in phase space. And this property of heart rate variability is something that underlies health. Now, a healthy subject has a chaotic pattern of heart rate variability, but it's not prominent. It doesn't overwhelm the statistics of the average heartbeat, which is what we focus on in terms of homeostasis. But it's absolutely essential for a sensitive heartbeat. And it's this kind of emergent property that underlies the quality of health that I'm going to keep coming back to over and over again. Fractals are indicative of something. They're telling us something about the system. Sometimes we understand it, sometimes we don't. But it shows up both in time, self-similarity in time, which is what this heart rate variability shows, and self-similarity in space, which is what you see in this kind of image. This, of course, is not real. This is a computer simulation of a fractal landscape, and intuitively you can see that you have, how clear that is, but you have mountains, hills, and you have structure on all scales. Most of the structures here are small, some are very large, and there's a very distinct relationship, frequency relationship, between the small and the large and those that occupy the space in between. And this is known as the power law. So the power law is something that captures a particular property of fractal systems. And I'm going to use it as an indicator of something deeper. It's part of the explicate order that's pointing to an implicate order that is taking us somewhere else. And I want to follow this scent, this hint, this suggestion in the direction of meaning. Here's another fractal structure. This again is a simulation. And it's very familiar. This is what a lot of foliage looks like. Plants have this self-similar structure. And it is, of course, maximally efficient at distributing fluids throughout a three-dimensional space to have a fractal structure. Hence, of course, a vascular system, the circulatory system, the way it branches, the trachea, the bronchi, these are all self-similar structures, and they're minimal energy configurations for doing a particular job. Distributing liquids, in the case of the heart, blood, or fluids, compressible fluids, like air, to the bronchi. And once again, we have this hint that there's something very deep about fractal structure. It gives coherence over a whole pattern. Here is the proteome of yeast. And what it's telling us is it's indicating the interaction pattern of proteins in yeast. And it's just giving you a diagram of what proteins interact with other proteins. And you can see that some proteins are very densely connected with others, but most of them interact with only one or two other proteins. And up in the top right, you will see the expression of the power law, which is to say, if you look at the probability of a protein interacting with k other proteins, and k takes a value 1, 2, 3, 4, up to 20 along the abscissa, and you plot that as a log plot, you get this straight line. This is a generic indicator of fractal structure. But the question is, what does it mean? What is it telling us? Now, I've used this term long-range order frequently to suggest that there is a kind of coherence that is being produced by a fractal structure. And coherence, of course, is an aspect of meaning. So I'm just tuning in on this structure as something that may be telling us something quite deep about what's going on in organisms and the patterns of interactions. So let me just go on to the next one. This is the yeast transcription network. This is the pattern of interaction for control of genes by transcription factors. And when you look at the distribution of the number of genes that are controlled by transcription factors, you find that the majority of transcription factors control only one other gene. Some control many other genes, and there's a power law distribution in between those. So that's the straight line here. There's a subset, which is the transcription factors that are specifically controlling themselves, transcription factors controlling the genes that are producing the transcription factors. So once again, power laws are emerging in these communication networks. Please interrupt me if something's not clear, because I'm going through this fairly quickly, and I want to remain coherent insofar as I can. Now, ambiguity in self-referential networks. Now I'm going to go into language. I'm going to talk about language. Because one of the empirical features of language, which was discovered by Zipf, who was in Harvard in 1949, published his paper saying that when you look at a text and you look at the rank order frequency of words in a text, it doesn't matter whether it's English or Spanish or any language whatsoever, what you find is that there is a power law description of the rank order frequencies of words in a text. That is to say, the most common word occurs with a certain frequency, the next most common word occurs with half of the frequency of the most common word, the third most common word is 1/3 of the frequency of the most common word, and so on. And this defines a power law relationship known as Zipf's law, ZIPF. So there's something in language that has this property of self-similarity and long-range order. And the question is, how does it come about? And I'm now going to refer to the work of Ferrer y Cancho and Ricard Soleil. Ricard Soleil is a colleague that I met at the Santa Fe Institute, with whom I wrote the book, whatever it's called, Signs of Life. Signs of Life. And Ricard has a research institute in Barcelona, and he looked with his colleagues at a model of the origin of language, because they were asking the question, why do we see Zipf's law of rank order frequency for words in a text? Where does that come from? So this is a model that attempts to explain the origin of this really quite deep property of language. Deep in the sense that you find it in all languages, but we don't know what it means. So, I'm going to go now into this area of exploring the meaning of Zipf's law in language and how we might understand that in relation to the evolution of language. What Ramon and Ricard did was to say, let's have a model in which we're looking at the evolution of language as a minimization process, the minimal effort between a speaker and a hearer who are trying to communicate with each other. Now, the minimal effort for the speaker is actually to use the same word over and over again, or word or sound, for all the objects in their universe of discourse. So just a sort of grunt. I know what I'm referring to, but you haven't a clue. So this is not an effective language. But it's one extreme, and children, very young children sometimes do this, to make a noise, and it's up to the parent to try to figure out the meaning of the noise. And so before there is articulated language, there can be various types of communication, but they're not very clear. So that's one extreme of this model. The other extreme is where every object in your universe of discourse has a specific word associated with it, and it's totally unambiguous. And that is machine language. That's the way we use language in machines, because we don't want any ambiguity there. Now, natural languages occupy a space in between these two extremes, in which you have ambiguity in the sense that a word spoken can mean many different things, and it could have many different acts of signification within the language. So there's ambiguity in spoken language. Now, what Ramon and Ricard did was to set up a function that minimized the effort for the speaker and the effort for the hearer, defined in terms of entropy functions, Shannon entropy. And what you then do is you minimize on both of these. So you're minimizing the effort for speaker and hearer. It's the minimum mutual effort. So it's a kind of compromise. And when you go through that operation for language, what you find out is that, and this is a graph in which, let me just go back to, there is a parameter in here, this lambda parameter. You adjust the lambda parameter to distribute the effort between the speaker and the hearer. And you're looking for a position of least effort that is for both speaker and hearer. And so on this graph, what we've got, the lambda parameter, it varies from 0 to 1, and it's across on the abscissa, and up the ordinate is the number of different words used in the language. And when you're minimizing the effort for the speaker, there's only one word being spoken. There's one signal that is used all the time, and that's totally ambiguous. At the other extreme, every single object in the world of discourse is specified by a word, and they're all used in this highly specific way, and that's machine language. there is another point of minimization. You have minimization for the speaker, minimization for the hearer. Minimization, the mutual effort, is actually right there in the middle. And that's a phase transition. And in the phase transition, what you find is Zipf's law as the distribution of rank order frequencies of words in the text. The most common word has a certain frequency. The next most common word has half that frequency. The next has 1/3 that frequency, and so on. And you get Zipf's law. So here's a model that suggests where Zipf's law might come from in terms of the evolution of language. Now, the results here are actually from a model that Philip Francis and I, Philip is a colleague at Schumacher College, and he did the modeling of this in relation to networks of control within cells, looking upon them as languages, communication networks. And so what we did was simply to take the model that Ramon and Ricard had produced for natural languages, produce it for any type of communication system in which you have a speaker and a hearer. And there's a sense in which this is what's happening in organisms all the time. We've got speakers and hearers, and we've got a process in which the organism is attempting to make sense of the information in the genetic text and the context of the organism, that is the external context, the cells with which, I'm thinking now embryologically or developmental, you think of developmental processes in which you have an organism going through a process of embryonic development, and it is generating a coherent order of a very complex kind, but it has this wonderful property of functional coherence, and at the same time, it has an aesthetic beauty about it. And so what I'm suggesting here, what Philip and I are suggesting, is that this principle of least effort also produces a structure, a morphology of maximum grace. Now I'm really pushing your boundaries. Use a word like grace. What does that mean? It's a kind of aesthetic coherence. And what you can see is I'm beginning to blur the boundary between what we call nature and the objective language of describing nature, and culture, which is the area where we are engaged in creative activity as humans. And what I'm suggesting then, this is another bit of data. Let me give you a bit more empirical data. This approach to the study of gene frequencies is something that Philip and I have applied to data coming from a laboratory in Bristol. We're collaborating, looking at microarray data from the superoptic nucleus of rats that have been dehydrated. And the whole question is, what is the pattern of gene frequencies when you have an organism that is presented with an issue, a problem, it's trying to make sense of its environment and do the most effective thing? And what we find out is that the pharmaceutical companies who were funding this research wanted to know which specific genes were they that were active so that you could design drugs to influence these genes. But it turns out there are not specific genes that are active. The coherence shows up across the whole of the genetic network. Thousands of genes in the microarray data, and they have a coherent pattern which is reflected in Zipf's law with a slope of -1. So this is just a little bit more data. So you can see the way we're thinking is to say, all right, the organism is presented with a problem, dehydration. What does it do? It looks for a coherent state to adapt physiologically to that condition and remain whole as an organism, whole and resilient and adaptable. And so that is then reflected, as we're suggesting. And so that is then reflected, as we're suggesting, in the power law relationship of gene transcription frequencies. This is a way of interpreting the complex data that's now coming out of patterns of gene action in organisms.